Okay, well, it seems to even out. So, hello everyone. Thank you so much for joining us today for the Huntington Society of Canada's monthly research spotlight webinar. So, at HSC, we're very grateful for the researchers at Canadian institutions whose creativity and their relentless determination are really driving progress in HD research all around the world, not just here in Canada. Today in particular, I think we're very fortunate to hear from a wonderful speaker. We have Rachel Harding with us today. So Dr. Harding's an assistant professor at the Department of Pharmacology and Toxicology at the University of Toronto, also a principal investigator at the Structural Genomics Consortium, also editor-in-chief at HD Buzz. So you have your hand in all sorts of different avenues of Huntington disease research and communications. And, I know your team also studies like the 3D shape of the Huntingtin protein and both its regular and disease forms, and with the goal to better understand the drivers of Huntington disease and then to find new avenues for therapeutic intervention. If during the presentation there's something that anyone in the audience would like more clarity on, if there's something they'd like to learn more about, we do encourage you to submit questions for Dr. Harding in the Q&A tab. So I'll be looking at these throughout the meeting and writing them down, and we'll aim to address as many of them as possible after the presentation. So thank you all so much for being here today, and over to you, Rachel.
Great. Thank you so much, Alexander. That's a very nice introduction. Yeah, it's my real pleasure to talk with you all today. So as Alexander mentioned, pile your questions into the Q&A, and then we can get to lots of discussion at the end, hopefully, if you've got anything that comes up. All right. So today I'm focusing on work that has been going on for a long time in both my lab and the lab of Cheryl Arrowsmith, who's also at the University of Toronto. And actually some of this work was funded by the HSC a little way back when we were trying to get this project off the ground. So we're always grateful for the funding that HSC provides to HD researchers in Canada. But what I'm going to be talking to you about is, we're really interested in studying the huntingtin protein, which, as you know, is the protein that gets changed in people who have Huntington's disease. And what we're interested in trying to find out, what does this protein do? How does it work? And how might we be able to target it for new ways to potentially treat Huntington's disease? But first off, I thought I'd introduce myself a little bit further. So yeah, as you can probably gather from my accent, I am not originally from Canada. I actually grew up in a small town called Dover in the UK, which is famous for its white cliffs and proximity to France. Although embarrassingly, my French is absolutely atrocious. So I do apologise to our folks from Quebec and other francophones who may be on the call. But I did my left there, and then I moved to Oxford to do both my undergraduate and PhD degrees before moving to Toronto to do my postdoctoral fellowship with Cheryl Arrowsmith. I thought I would stay for maybe a year or two, but absolutely fell in love with being in Canada, and I love Toronto. It's an amazing city for research and a wonderful place to live. And I've basically been here ever since. So after I finished my postdoc, I started my independent research career, and I now have my own lab at the University of Toronto, and I'm also co-editor-in-chief of HD Buzz. So I always like to flag HD Buzz up front. This is a news website for all things Huntington's disease, really focusing on the latest research and clinical trials. And together with my good friend and colleague, Sarah Hernandez. Oh my gosh. Hang on. Can you still see my full screen? I'm seeing a funny thing on my screen.
It kind of went down into like a little corner, but now it's back.
Is it back to full screen? Okay. All right, I don't know. Let me know that you can definitely still see it, and then I can carry on. Okay, because I'm going to not touch my mouse because clearly there's something. I've set in some spooky settings here. But yes, back to HD Buzz. So this is where you can find out everything that's going on in HD research. We try and provide, really up to the minute updates on all the latest clinical trial news. And you can follow along. Like, one of the best ways to do this is you can actually scan the QR code on this slide, and you'll find that you can read all about it will take you through so you can sign up with your email. So you get all the news delivered to your inbox and you can find out what's going on. The other thing I wanted to point out is if you go onto our website now, we have this thing called the trial tracker, which basically there are so many clinical trials in HD right now. It's kind of hard to keep track of who's doing what, when, and when we're expecting the latest news. So this is a good way to kind of see like what is coming down the pipeline and what we should, which companies we should be hearing from when. So we do our best to keep this up to date and accurate. So yeah, I recommend checking that out. Okay, back to research. So yeah, as Alex mentioned, like we're not only based at the University of Toronto, but everyone in my team is also a member of an organization called the SGC, or Structural Genomics Consortium. And so our lab is, we're split across two different places. And the reason I mention this is because the SGC is this quite interesting organization. It's a public-private partnership and it's entirely focused on open drug discovery. So that means that every single thing that anyone in my lab does will go eventually into the public domain. We share all of our research with no patent restrictions, no restrictions. And this is because we really believe that this is a way to accelerate research by sharing our materials and knowledge with people as quickly as possible so that we can just get science done and we're not wasting time with boring administrative agreements and so forth to protect IP, which is normally not worth it. But the SGC overall is like we're really focused in these areas of sort of understudied drug discovery. And we have sites all around the world. The biggest one is in Toronto at the UHN hospital system. We're based in Princess Margaret Cancer Hospital. But we have sites in Frankfurt, in Sweden, in North Carolina, in Brazil, all over the place. And there's hundreds of us scientists all working on these interesting and understudied areas of disease research. And we collaborate with people all around the world. And you might be like, why am I telling you this? And it will become important later when we talk about some of the projects that my lab is running and how this collaborative spirit has been like really critical for us to having some success in our HD research programs. But what we're fundamentally interested in is really understanding biology at this kind of detailed molecular level. And here I'm showing you on the slide, a person and then a brain and then a neuron and then a protein molecule. And we're interested in these protein molecules, all these molecules in biology that do lots of really cool things. These are also very often, the molecules that are going wrong when we have disease and when things aren't working so great. And one thing that I think is really interesting is thinking about the scale of what we're trying to look at. So, if I made this neuron, which is 20 micrometers, as long as I am tall, a protein molecule would only be the thickness of my fingernail. So these are really, really tiny, tiny things that we're trying to study and really understand at a very detailed atomic view. And these are proteins that do all kinds of different jobs. You will know about many of these. So it's about our immunity, our hormones, how we digest our food, our hair and our nails, and all sorts of different things. But in the Harding lab, what we're interested in is proteins that are relevant to Huntington's disease, and in particular, this protein called huntingtin. And we're interested in understanding, how does this protein work? How has it changed in Huntington's disease? What can that tell us about new avenues for possible drug discovery for HD? And we're also interested in diseases that are a bit like HD. They come from similar kinds of genetic changes, and what those might be able to tell us about our HD research programs as well. And as I mentioned, everything goes into the public domain so that we can share things with everyone around the world. So let's start with a bit of HD Biology 101. And for those of you who've seen my HD Buzz presentations before, I'm sure this is excellent revision around everything that underlies Huntington's disease. And so what we know to be true is that most people with Huntington's disease make two different types of the huntingtin protein. And this is because we all have two copies, or they're sometimes called alleles, of the huntingtin gene, one from our mum and one from our dad. And so most people who have Huntington's disease will make both the disease form of the gene and they'll have the healthy form of the gene as well. And this means they make both the disease and the healthy form of the protein. So what do we mean by that? Well, what happens in the Huntington gene is that even in folks who don't have Huntington's disease, there's this run of DNA letters that say CAG, CAG, CAG over and over and over again. And in folks who don't get Huntington's disease, they'll probably have anywhere from 10 to 26 of these CAG repeats. But people who have more than 40 will go on to have Huntington's disease in their lifetime. And then there's this kind of gray area in between where if you have 27 to 35, like your children may go on to develop HD. It's quite unlikely that you would. But if you have 36 to 39, you may or may not get Huntington's within your lifetime. And what this genetic letter change does at a protein level is it means that we make a different version of the Huntington protein. And so for folks who have the unexpanded Huntington gene, they will make the regular kind of what's sometimes called the wild type Huntington protein. And we know this goes on to, there shouldn't really be any problems with nerve cells and the symptoms. We shouldn't see any symptoms in people with this kind of Huntington gene. But with folks with HD, they have this expanded Huntington gene. They make an expanded form of the protein. Sometimes this is called the mutant Huntington or mHTT protein, but we prefer expanded. And this can make our nerve cells in our brains sick. And this is kind of what starts the symptoms that we know people with Huntington's disease experience. But the thing my lab is really interested in is trying to figure out like where is this happening? And it turns out this CAG repeat or the CAG number encodes for an amino acid called glutamine. And so because we have repeating CAGs, this means we get repeating glutamines. And this is right at the very, very beginning of the protein molecule. And this really matters because Huntington is this enormous protein. It's actually made up of more than 3,000 amino acids in one really big long string. And we have these glutamines at the very beginning, which get changed. But overall, so it's not like a huge change in, the overall structure of the protein remains exactly the same. The overall coding from these amino acids stays the same. It's only this little bit at the very beginning that has changed in people with HD. And to try and understand, what this change in the stretch might mean, so we have to think about, well, what does the protein do normally? And one of the ways that we like to think about the Huntington protein is it's basically like a Swiss Army knife in the cell. It has lots and lots of different jobs. It seems to be important for how nerve cells carry messages, how we look after our DNA in cells, how we send different protein molecules to the cell trash can, and all sorts of different things. And you can think, well, how can one molecule do all these different jobs? This is quite different to when we're thinking about the examples we thought about earlier, where we have, these enzymes that help us digest our food. They're not doing loads of other things as well. That's just one job. Whereas Huntington has all these different jobs. And that's actually, for someone like me, that's something that makes it really exciting to understand and try and figure out what's going on. And we think it's able to do all these different jobs because it basically acts as like a kind of landing platform for all of these other proteins in the cell to jump onto. And all these other proteins in the cell do lots of different jobs. So depending on which other proteins jump onto Huntington, it's probably going to do different things. As we call this is like, think of Huntington as almost like the scaffold or the frame for all these different molecular machines that are going to work in the cell. But this is, if you're thinking this sounds like a bit of a vague explanation, you are absolutely correct because this is not really detailed. We don't know all the molecular underpinnings of exactly what Huntington is doing. And I think this is really important because, there are still so many things in the field where we're really thinking about all these basic, what I would think of pretty basic questions to try and understand the Huntington protein. So what is its normal job? We know it's a scaffold, but what else? What does that really mean? Where do we find Huntington in cells? Which type of Huntington matters most in disease? Like we talk sometimes about these aggregates or toxic protein clumps, but we also have like the regular protein as well. Like how are these all working together? And exactly how does this change at the beginning of the Huntington protein really impact, disease? Like how do we go from this small genetic change to a small change in a protein to suddenly having what we know to be this really devastating illness? And I think the answers to that thus far, in my opinion, remain a little bit unsatisfying, and there's like a lot more work to be done. But a fundamental truth of Huntington's is that we know, depending on how many CAGs you have, you will or will not go on to have Huntington's disease and all the symptoms. And so one way that we can think about designing therapeutics is like, well, let's just switch off or make less of this expanded form of the Huntington protein. And this is what a lot of companies are doing now in the clinic. These are called Huntington lowering therapies. And if you look at this really nice model of like how all the different companies who are either in the clinic or on their way to the clinic, you can see under Huntington lowering, there are all these different folks, which is so exciting. Like all these people working on Huntington's disease. And these aren't just, small biotechs. These are massive companies like Novartis and Pfizer and all sorts of folks. So there's huge interest in developing drugs for HD. But most people are taking this approach, like sticking with that fundamental truth of will making less of the expanded Huntington protein help improve symptoms? And the honest answer is we don't know whether that's true yet, but I really hope it is true because that would be amazing. So this is what everyone's doing in the clinic, but what we're thinking about is trying to understand this at a more detailed level. We still have all these questions about Huntington protein. We're now in the clinic making less of the Huntington protein. So it seems important to me that we really understand what are the potential sort of downstream effects of these drugs at a detailed molecular level. And that's what my lab is interested in doing. And so what we need are new ways to study this Huntington protein so we can really dive in to a lot of the detail about how this molecule is shaped, what's its 3D shape and structure, how does it work, and like what can we try and figure out about this protein. So the way we do this at the SGC, as I mentioned, is that we are really interested in understanding these protein molecules at a detailed level. If you want to get any experiments done on a protein, first you've got to be able to make that protein in the lab. We would never get ethics approval to start, taking pieces of human and trying to extract Huntington protein out of that. That is not how we do science these days. And so what we need to do is find a way to, make protein factories in a lab setting so that we can just make as much Huntington as possible. And so actually, it's quite interesting. We actually do this in a type of insect cells. Insect cells, you can grow them in these big flasks, and we can make a lot of biomass from which we can basically genetically engineer them to become Huntington protein factories. And we can make huge amounts of protein. And once we've made the protein, we have to purify it. So we end up with a sample that's really clean. It's like a nice sample. It's like folded properly. It's very pure, and we can make as much as we're going to need to. So we call this scalable production. And one of the good things about this system is that it's essentially enables us to make lots of different flavors of the Huntington protein. So we can make Huntington with all kinds of different polyglutamine lengths, which either mimic people who don't have disease or people who do have the Huntington's disease gene. When we're trying to understand this molecule, or any molecule, what we really want to know is like, what are the details of like how this molecule is working? What are the details of how it is shaped? And so you can kind of think of this as like all proteins are kind of cellular tools. They're doing all these different jobs in a cell. And in the same way, if you were reaching for two tools from your toolbox and you only had an approximate idea of their shape, or it would be quite hard for you to look at them and know what they were doing. So if I asked you, what are the tools behind these green and blue crosses, and what is the role of these tools, it'd be quite hard for you to answer without seeing the details. But when I remove them, you can see like, although these are quite similarly shaped, they're actually two tools that do quite different things. So we go pliers and we have cutters. And so we can now see that even though they look quite similar, we can see that they do two very different things. And this is what I do and what my team does as structural biologists is we, instead of looking at tools, we look at protein molecules to understand what they might be doing. And so here with our really nice Huntington protein samples, we're able to use special microscopes to start zooming in to look at the overall shape and structure of the Huntington protein. And so what you can see in the bottom left is this is what the Huntington protein looks like. It's quite a funny looking molecule. It's got this big hole in the middle, and it's kind of like, this C shape with the top and the bottom being this purple and blue bits connected by the green in the middle. And you would not be be wrong if you thought, well, this looks quite blobby and I can't really look at this and understand what it's doing, Rachel. So can you not do anything better in the lab? And you'd be spot on and absolutely correct. What we really need to do is to try and bring things into focus so we can see more clearly how are all the atoms arranged, how do they connect with each other, and what does that tell us about how the protein might be working. And so it turns out huntingtin has a really good friend called HAP40 that it likes to stick around with. And now when we look at huntingtin under the microscope, but we've got HAP40 there as well, which is this yellow piece in the middle, you can see that all of a sudden, all of this detail comes into play. So we can see really nice definition of the molecule and how all the atoms are arranged in three-dimensional space. So this is really cool, and it tells us that huntingtin obviously really likes being around the HAP40 protein, and this is a really important binding partner. And so now we've got these samples which we understand, we can look at their structure, we can make a lot of them, we know that they're high quality. One of the things my lab does is that we provide this protein to folks all around the world. And so because we're not restricted by any IP agreements or any material transfer agreements or all these other boring pieces of red tape that the universities like to make us do, we will just send protein to any lab around the world who is interested in studying the huntingtin protein. And this means that there are, we think we've got lots of really good ideas for what to study for huntingtin, but I can tell you there are lots of really clever people out there who have all kinds of really interesting ideas for what they want to do and how they think the huntingtin protein might be working. And so we've actually shared materials with more than 30 research groups all around the world now. This is hundreds and hundreds of aliquots of prepared sample. And there's been, some things have worked out really well, and there's some exciting examples of collaborations that have come from this. We have, well, two here that I want to highlight that are both in Canada. So Francesca's lab at the University of Laval, we were looking at, are there antibodies, does your immune system have a reaction to the huntingtin protein in people with Huntington's disease? And we found that we could see that there are antibodies that actually target huntingtin in people with Huntington's disease. Really interesting. We also work with Ray Truant on understanding how huntingtin might be working in DNA repair. And there's another nice example here with Amber Southwell, where here we're looking at how we can measure huntingtin accurately. This is really important for all those huntingtin lowering therapies I was telling you about earlier. We want to make sure our drugs are doing exactly what we designed them to do. Are they lowering huntingtin protein? And all of this was enabled by the clever people in my team who make all this protein. They share it with people. We have an enormous FedEx shipping bill, but it's all worth it because we get to do all of this fun research with people all over the world, which is super awesome. So this is great. We can apply huntingtin protein to these different settings. But what about if we want to study huntingtin in the cell? What we really need is a way to kind of fish it out of different types of cells and kind of hook it and see what's going on. Like, what is it up to when it's in the cell working, doing all sorts of different things? And for this, what we really need is, we call these chemical tools. So unfortunately, there is not a fish hook that is designed to be small enough to work on a huntingtin protein molecule, but there are other types of hook that we can use. And one of the things that is interesting about huntingtin is it's so big, and actually designing a tiny helpful hook to try and hook out our really big huntingtin protein is not always that trivial. So we decided to make it easier, and we used a big hook to get our big huntingtin protein, which is displayed as this large fish in my rather clumsy analogy. So what we use is this kind of larger kind of drug molecule. So this is something that's called a macrocyclic peptide, which is basically just like a big word for lots of amino acids pulled together into a string that makes a circle. So, this is very similar to how the proteins in our bodies are made, but this time we're making them much shorter, and they go round in a circle instead of being a long continuous string. And this is a cool idea that lots of people in different types of drug discovery are doing. And there's all kinds of partnerships of like how, with Merck and Pfizer and Novartis, all these companies are really interested in this new approach. And so we actually ended up working with one of the inventors of the original technology, who's this guy called Hiro Suga, who's at the University of Tokyo. And we sent him, he's one of the people that we sent our huntingtin protein to. And really, really quickly, he sent us back a ton of data and was like, great news. It looks like we found some binders for your huntingtin protein. So these are our fish hooks to try and study the huntingtin protein. And so you can see there's 5 different ones that I'm showing you here. I'm just going to talk about the one that is highlighted on the left here in purple, just to walk you through some of the things that we can do with this kind of tool. So before using any tool, we want to be really sure it's doing what we designed it to do. And so Esther, who is postdoc in my team, she designed all these different kinds of experiments to measure how well does this hook on to huntingtin. And so she did the experiments you can see on the left and the right side panel. These are called surface plasma resonance and fluorescence polarization displacement assays, which is just lots of language for 2 different ways that we can measure the hooking effect of these molecules on the huntingtin protein. And the good news is that basically the lower the number, the better. And you can see that the values here are very low. So we have 2.2 nanomolar binding, which is very, very tight. So this means the hooks are working really, really well with the protein that we can make in the lab. We then want to see, like, well, where are these hooking onto huntingtin? And for this, we use another method that's called HDXMS. This is hydrogen deuterium exchange mass spectrometry, which is a bit of a mouthful, but essentially this is a bit like of a way of footprinting exactly where your hook is hooking onto your protein. And you can think of this like if you park your car and then it snows, when you drive away, you can see exactly where your car is, or where your car was. And that's what we can do with these drug molecules. We can basically see a footprint of where they are on the protein molecule and identify these. And this is really, really helpful information because then we come back and we use those powerful microscopes again to see exactly how everything is binding on. So this is like getting out, our binoculars to really zoom in and see all of the details of which atoms are close by to each other. How is our hook contacting the huntingtin protein? So now with all that information, this is really helpful because this is a bit like if you have, we now have, it's almost to jump analogies even further. So I apologize again for slamming together too many analogies in this talk, but you can also think of many of these molecules are like being, a key for a lock. But with this kind of detailed information, it's like having a clear padlock where you can see inside and you can see exactly how all of these things are working. So for people like me who are drug designers and people who understand protein structure, this is really, really valuable information for us to move forward with. And then Rebecca, who is a PhD student in my team, she is the one who did the first actual huntingtin hooking experiment. So she took these tools and she was able to pull out huntingtin from cells. And you can see on the right-hand side here that she can see both the huntingtin and its friend, HAP40, have come out when she hooks huntingtin out of cells. So this was really exciting. We've never done this before. And we were so over the moon with how well this is working. We went on and actually this was a really nice collaboration with Mahmoud Puladi's lab. So he's at UBC and he basically gave us samples of different types of cells that are either from regular folks or people who have Huntington's disease. And you can see that it didn't really matter. We could always pull out the huntingtin protein with these new tools. So that was really, really good because it means that we can apply them into different disease models moving forward, which is pretty cool. Okay. So this is all, oh yeah, and now I highlight it for you here. So this is all well and good, but these big hooks are big and clunky. And there's some experiments we can do with them, but if we're thinking about how can we use this information to design things that will, bind onto the huntingtin protein and could be potential starting points for different therapeutics or drug discovery, we want to shrink these all the way down. And we want to make something which is called a small molecule. So this is something which is much, much smaller than these macrocycles I was just telling you about, but is going to have much better likelihood of being able to get, into the CNS or get into the nervous system if we turn it into a drug or all kinds of different things. They're just easier to use, easier to handle. The problem is that huntingtin is so big, trying to measure these kind of small molecules binding to huntingtin is just really, really challenging. And so we need really, really sensitive biophysical assays to do this. And so these are different ways that we can measure how a small molecule might bind onto our huntingtin protein. And so I kind of think of this as like the princess and the pea. Like we need to find our princess, someone who is so sensitive that through all the layers of a mattress, you can still feel a pea. We basically need a biophysical experiment in the lab that will do the same thing. It's super, super, super sensitive. And so we're really lucky, with this with our partnership with the SGC, that we're able to do some of these things. So here, what we're able to do is we want to try and find these small molecules that bind the huntingtin protein. They have a platform that's called affinity selection mass spectrometry, or ASMS. And as you're probably learning from this talk, scientists like to have lots of complicated names and big acronyms. And essentially what we're able to do here is we're able to hook out, now we use our huntingtin as the hook to see, can we fish out different small molecules that will bind to our huntingtin protein. And we're able to do this pretty quickly. And this is kind of like a really clever way to detect these tiny, tiny things, like using a metal detector to detect a needle in a haystack. And then we were able to go on and use all those approaches that we thought about earlier with like, how can we measure these are binding to our huntingtin protein? We were able to do that with our macrocycles. How about with our small drug-like molecules? And so Rochelle, who is another PhD student in my group, she spent a lot of time and energy getting this really well thought out and organized. And she was able to show that one of the small molecules that we found with our metal detector in the haystack, she was able to measure the binding of this to the huntingtin protein. And this was like so exciting. I think she is the first person to have done this for the huntingtin protein ever, to the best of my knowledge. So really incredible data and such a breakthrough, in my opinion. And so then we were able to go on with all these things that we talked about earlier. We did our, car park in the snow trick to try and work out where is this particular molecule binding on the huntingtin protein. We're able to map it out. What's really cool is that we had previously predicted that huntingtin could potentially be drugs at this site all the way back in a study in 2021. So this was one of the only times in my life where I've seen a prediction that I've made in a paper come true a few years later. So that was a nice turn of events. And then we did the same thing where we used our powerful microscopes to really dive in to see exactly how is this molecule binding onto the huntingtin protein as well. And this is really cool because now we're thinking about what can we do with these? So right now you can look at all of the different ways that people are doing huntingtin lowering in the clinic, and a lot of this is quite difficult for people who want to participate in trials. Either they've got to have injections in their spine, or they've got to have a brain surgery if that's what you're participating in the Unicure trial, or it's just not trivial, like how we're going to get some of these drugs delivered to people. But what we're hopeful for is with small molecules like this, we can actually turn these into molecules that can degrade the huntingtin protein directly. And so this means that we can actually make this molecule slightly bigger, and it will end up recruiting different types of cell machinery to send huntingtin to the trash can. And you can actually see there are a few different companies doing this. This is called protein degraders. You can see it on the right-hand side of the slide here. And so this is like a really cool area that we hope will, develop over time, and is something my lab is really interested in doing. The other thing is that we don't have a good way right now of knowing, is huntingtin being lowered in the brains of people who are receiving different huntingtin lowering drugs. Instead, what we're doing is we measure the spinal fluid that bathes the brain to see how much huntingtin is there, and does it change if we give someone the drug. And this is a good proxy. It gives us a good estimate of how well the drugs are working, but it doesn't tell us where the huntingtin lowering is happening. And so this is one of the questions that came up with the recent data and the recent announcements from Roche, was like, what is happening with tominersen? Is it lowering huntingtin in the right places that we really care about for Huntington's disease? And what we could do is, again, we can go back to this molecule. There's the potential to turn this into something that we call a PET ligand, which means that we could if we're able to do that, we could administer it to people and it will light up parts of the brain where there is the huntingtin protein. And then we can directly measure in living people how much huntingtin is left. So that would be super cool. And these are all things that my team are really interested in and we're trying to push forward. And yeah, so I think I'll leave it there. The key message here is like, we really think that these kind of public domain tools are going to be important for unlocking biological discoveries and also developing new therapeutics. And everything that is done in our lab is open source and available to the research community. So all of this work is, I've gone over a lot of stuff very, very quickly, but this is a huge amount of work from people in my team, past and present, as well as huge support from the Structural Genomics Consortium team at the University of Toronto and all of our collaborators as well. And you can see quite a few Canadian labs in there, which is pretty amazing. I feel like Canada always punches above its weight for HD research, and that's something that we should all be very proud of. I'm also very grateful for all of our funders who help us get all this work done. And if you have any questions, maybe you don't want to ask in this particular call, that's totally fine. You can also just drop me an email. And we also host lab tours for folks in the HSC community as well. So if you're interested to come and see how the sausage is made, so to speak, in our lab, you can also shoot me an email. We'd be delighted to host you.